Lately, we've been focusing a lot on the DNA of a cell. And so if you're considering a eukaryotic cell, remember that where you'd find its DNA is inside of its nucleus. We've said that basically a cell does two things with its DNA. One of the things is this. So if you consider a chromosome, remember that a chromosome has several hundred genes on there. And what each gene does is that it serves as a set of instructions that tells a cell how to make a particular type of protein. And so if you have different genes, then that different gene will tell the cell how to make a different type of protein. Another thing that we saw DNA doing is that a cell can copy it. And the reason why a cell would copy its DNA is so that its genetic material can be passed on to any daughter cells that it produces. So again, basically a cell does two things with its DNA. One is that it can copy it, and we saw how cells copy its DNA last week. And another thing that cells do with their DNA is that they use the genes on the DNA as a set of instructions that tells a cell how to make a particular type of protein. And so that's actually what we're going to look at here. So how does a cell read the information on DNA in order to make a type of protein? Before we take a look at the details of that, how about if we look at kind of a big picture of how a cell makes proteins? So you might remember way back in the first week we showed you this picture, and this is basically showing you what a cell does with its DNA. It uses it in order to make proteins. Again, if you're talking about a eukaryotic cell, remember that its DNA is safely tucked away inside of the nucleus. The place where the proteins are made, though, are way outside here in the cytoplasm at the ribosomes. So we mentioned back then that in order for the information to get from here to out here, it turns out that the DNA always in a eukaryotic cell kind of stays safe inside the nucleus. So in order to get the genetic information from here to here, basically a cheap copy of the genetic material is made, and that cheap copy is in the form of a messenger RNA. And it's that RNA that then leaves the nucleus to travel to the ribosome, and that's actually where the information on that genetic material is read in order to make a string of amino acids or a protein. So it turns out that the steps that you see here in making a protein, they're called something. And so here's what they're called. So this first part of making a protein, where the DNA is read by a cell in order to make kind of a temporary temporary molecule that holds genetic information. Again, that molecule is called a messenger RNA. That process where DNA is read in order to make messenger RNA, that's called transcription. Right, so transcription is making RNA using a DNA template. And this might make sense that this process is called transcription, because if you think of what happens whenever somebody transcribes something, transcribing something just means that you're copying it. And the same thing is happening here. When DNA is used to make RNA, the genetic information is just being copied, right, from one nucleic acid to another. So that's why it makes sense that this process of making RNA from DNA is called transcription. So the reason, again, for making that RNA is because it's the holder of genetic information that actually leaves the nucleus to go to the ribosome, where the cell now can directly read this in order to make a protein. And again, remember, proteins are just a string of amino acids. So this process that you just saw here, where the messenger RNA is used to make a protein, that's called translation. And so it makes sense that this process is called translation, because if you think about what people do if they're translating, for example, a language, right? They're switching from one language into a completely different language. And so the same thing is happening here. Information is being completely translated from one form to another. So you see that you start, during translation, you start with a nucleic acid, and at the end you end up with a completely different molecule, a protein. So the information is being translated from one molecule, nucleic acid, to a completely different one, protein. That's why it's called translation. So again, basically making a protein in a cell involves two steps: transcription, where the DNA is used to make RNA, and then translation, where the RNA is used to make finally the protein. And so we'll take a look at the details of transcription first, and then later we'll take a look at the details of how translation works. One thing to take a look at though or one thing to note before we look at the details of transcription is this, kind of another big picture concept, is that if you consider a chromosome, again, chromosomes are made of DNA. If you consider a chromosome inside of a cell, each chromosome actually has hundreds of genes on there. And so this means that each chromosome can be transcribed many different times, right? Because it has many different genes. So each chromosome has the information to make several different types of messenger RNAs. And it's each messenger RNA then that goes to the ribosome in order to be read in order to make a protein. We're seeing here that if you consider all the genes on a chromosome, only some of them at any one time are actively being expressed. In other words, only some of them are actively being transcribed to make messenger RNAs, and only some of those are actively being translated to make proteins. And so next week we'll take a look at how a cell can turn off genes. So for example, here genes 2, 3, 5, and 7 are turned off. They're not actively being expressed. And so again, next week we'll take a look at how a cell does that. But for now, we'll take a look at how this process occurs. So if a gene is turned on, how is this process of transcription, how does that occur? And then if a gene is turned on, how does this process of translation from messenger RNA to protein, how does that occur? So again, first we'll take a look at how does this happen. How does the cell read the information on a DNA in order to make a messenger RNA? So say that here is a gene on a DNA molecule. And again, the goal of transcription is to read this information and make a messenger RNA out of it. What we'll mention right up front is that you'll see that this process of transcription, it looks very similar to the process of DNA replication that we took a look at last week. And the reason why is because during DNA replication, basically a DNA molecule is read in order to make another DNA molecule, another nucleic acid. Well, basically the same thing is happening here. A nucleic acid is being read in order to make another type of nucleic acid. So that's why, again, transcription is going to look very similar to just plain old DNA replication. And so if you remember what has to happen first in order to read the information on this DNA, well, the information is locked up in the middle here in these nitrogenous bases. So in order to get to that information and read it, the two strands have to become separated from one another.